Good morning, guys. I'm Chris Mathew from MSc Biotechnology from Krista Jayanti College, and I'm doing my activity-based evaluation, which is presenting a seminar which is submitted to Dr. Manikandan Kathirvel, and the topic is blotting techniques, and I'll be specially talking about southern and northern blotting, its principle, procedure, and application. As an introduction, I would like to say what bloating is. Bloating describes the mobilization of a sample nucleic acid or protein onto a solid support, and you transfer this nucleic acid or protein to a chemically reactive matrix, which is called a bloating membrane, which is typically nitrocellulose or activated nylon. It can be charged, positive, negative, or uncharged. And this protein that is immobilized into the membrane can be a pattern similar to the one that is present on the original gel itself. It is usually carried out after the gel electrophoresis separation of the compound. When the bloating procedure is done, the transferred protein or nucleic acid is visualized usually by color staining, that is has an example, silver staining for proteins, or it can be autoradiographic visualization, which involves specific labeling, like proteins can be labeled during cell growth by incorporating amino acid containing different isotopes, or it can be a hybridization probe or antibodies. And the enzymatic activity is visualized by incubating it in a proper reactivity after proper washing, giving it either a color deposit or a chemiluminescent reaction. Now, this is a tabular column which shows the different bloating membranes used for different nucleic acids. And the bloating techniques are of several types: southern for DNA, northern for RNA, western for proteins, eastern for post-transcription translational protein modifications, and southwestern for DNA or DNA protein interactions. Now, this is a typical capillary bloating apparatus of bloating. Now, the gel is placed on a filter paper wick, and a nitrocellulose paper or nylon membrane is placed on top. Further sheets of the filter paper and paper tissues complete the setup. Transfer buffer is drawn through the gel by capillary action, and the nucleic acid fragments are transferred out of the gel onto the membrane. The plastic tray contains the transfer buffer. Now, southern blotting. It's a technique which was invented by Edward M. Southern in 1975. Southern blotting is a principle. Southern blotting has a principle that uses the separation of DNA fragments with gel electrophoresis and then identified by a labeled probe through hybridization. It is an example for RFLP, and it reveals the information on DNA identity, size, and abundance. So the principle itself is hybridization, which refers to the process of forming a double-stranded DNA between a single-stranded DNA probe and a single-stranded desired target DNA. Now, since the probe and the target DNA are complementary to each other, the reaction that occurs is also specific. Now, for a sufficient southern blotting to occur, a large DNA fragment less than 10 kb is required for a proper transfer in a short time, and hence it should be a short fragment. To allow uniform transfer, a wide range of DNA fragment sizes should be present, which is electrophoresis, and then treated for depurination by alkali, which can be 0.25 molar per liter HCl. Now, this shortens the DNA fragment through alkaline hydrolysis, that is the depurination, and then a positively charged nylon membrane can be used to hybridize it. Nucleic acids, especially RNA, can be used for UV radiation for 5 minutes, which is the UV allows the covalent linking between the T residues in the DNA and the positively charged amino groups on the surface of the nylon membrane. Now, the steps of southern blotting. The first step is the extraction and purification of the DNA from cells, which involves the lysis of the entire cell, and then the use of proteinases to remove protein, and then it is purified by alcohol precipitation, and fibers are removed using buffer. The second step is restriction digestion, where we use restriction enzyme, and the DNA is cut into several fragments and then multiplied by polymerized chain reaction. The third step is separation by electrophoresis, that uses agarose gel electrophoresis, in which the negatively charged DNA moves towards the positively charged anode in a distance that moves depends upon its size. The depurination is then done next, which involves the use of diluted HCl, which has high efficiency transfer of DNA fragments and breaks them down into smaller pieces. Then the denaturation, which involves mild alkali, with that is such as NaOH, that can cause a double-stranded DNA to become single-stranded and making it suitable for hybridization. And then the DNA is neutralized with NaCl to prevent rehybridization before addition of the probes. Now the blotting occurs where it's transferred onto a nylon or nitrocellulose filter membrane, and then finally some dry filter papers are placed on top of the membrane, and the fragments are pulled towards the nitrocellulose filter through capillary action, as I explained before with the diagrammatic representation. The seventh step is baking and blocking. So we cannot use UV cross-link here. We cannot use UV for cross-linking between the molecule and the membrane because of the DNA is not suitable for that. So we need to bake it in a regular oven or a vacuum at 80 degrees Celsius for 2 to 3 hours, and then we add casein or bovine serum albumin, which saturates it. Now the hybridization with the labeled probes occur, where it is treated with the labeled probe and which contains a complementary sequence of the gene of interest to the target DNA sequence, and the probe binds with the complementary DNA, as I already said, with non-specific binding sites on the membrane, which is blocked by BAC and casein, so it only binds to the specific site. Now, washing of the unbound probe using buffer occurs as the next step, and then it's visualized by an autoradiogram, or it can also be used for visualizing. We can use X-ray films or phosphorimaging instruments, or it can be while it's incubated with the chemiluminescent substrate, the color can be observed or blotted by an X-ray film, and detection time is around 1 to 48 hours. That's 1 to 2 days, 1 hour to 2 days, and it depends mainly on the probe. Next are the applications of southern blotting and a small diagrammatic representation. So as I said, it is used as a preparation for RFLP maps. It is specific for DNA in the DNA samples, and the detection of mutation, deletion of gene rearrangement in DNA can be seen. It is used for criminal identifications and DNA fingerprinting and so on. Next is northern blotting, which was developed by James Alvin, David Kemp, and George Stark at Stanford University with the contributions of Gerard Hendrich in 1977. It is the method that is involved for RNA and instead of DNA. And this technique is used for gene expression, and it is done by detection of a particular RNA or the isolated mRNA, which is almost the 5% of overall RNA sequences. This method reveals the identity, the number, activity, and size of a particular gene. Now, as a principle, I would like to talk about what the probes actually are. So the probes are nothing but the nucleic acid with complementary sequence to all or parts of the RNA of interest, and it is oligonucleotides with a minimum of 25 complementary bases to the target sequence. Now, the steps of northern blotting. First, the cell is lysis. Then the inhibition of the ribonuclease activity occurs. Then depurination occurs and the recovery of the intact RNA. Now, the mRNA is isolated by oligo-DT cellulose chromatography, where the oligo complementary to the poly A tail is covalently attached to the resin column. And then after the sample has been applied, the mRNA that has the mRNA, the poly A tail will hybridize to it, and then after elution buffer is applied, the disrupt hybridization, and then the recovery of the mRNA occurs. The next step is probe generation, which involves a northern blot that can be probed with radioactively or non-isotopically labeled RNA, oligonucleotide probes, and this has 10-fold of sensitivity. Then it is denaturation with the agarose gel electrophoresis. So step 3 was the denaturation with the agarose gel electrophoresis. The agarose gel contains formaldehyde, which is traditionally used for denaturing RNA because the formaldehyde reacts with the imine and amine groups on the nucleic acid, which disrupts the hydrogen bonding between bases and disrupts the secondary structure of the RNA. It is important to disrupt the secondary structure because the RNA must be extended to allow proper binding of probe for identification. Polyacrylamide gel electrophoresis unit with urea has a denaturing agent and also can be used, and ethylene bromide in turn can be used as a staining agent. Now, step 4 is a transfer to a solid support and immobilization. Now, the separated RNA sequence is transferred to the nylon membrane. This is done by two mechanisms, that is capillary action and the ionic interaction. So, the transfer operation is done by keeping the gel in the following order. First, the agarose gel is placed on the bottom of the stack, followed by the blotting membrane. On top of this, are paper towels of mild weight and a glass plate is placed. The entire setup is kept in a beaker containing the transfer buffer, which is the diagrammatic representation I showed in the beginning, that is the capillary action of bloating. The transfer is necessary because the probe can't enter into the gel matrix. Therefore, the RNA must be transferred to a membrane where they can be assessed by or accessed by the probes. Nylon is commonly used, and the bloating membrane is positively charged to attract the negatively charged RNA. Transfer is accomplished via capillary action, as I said, and or by electrobloating, that is around 15 to 60 minutes, 15 minutes to 1 hour in a blotting system. Step 5, that is pre-hybridization and hybridization with probe. The pre-hybridization or blocking requires prior to probe hybridization to prevent the probe from coating the membrane, and good blocking is necessary to minimize the background problems. RNA transfer to the nylon membrane is then fixed using UV radiation. Short-wave UV lights causes nitrogen bases in RNA, mostly uracil, to become highly reactive and to form covalent linkage to the amine groups on the surface of the membrane. And damp or wet membranes require an exposure of approximately 120 millijoules per centimeter square. The fixed nylon membrane is then mixed with probes, which is specifically designed for the gene of interest that will hybridize with the RNA sequence and the corresponding to the sequence of interest. The next step, the sixth step, is washing. After hybridization, the unhybridized probes are removed with washing with several buffers. The low stringency washes are done with 2x SSE or SSPE. Remove the hybridization solution and unhybridized probes. High stringency probes that involves 0.1x SSE or SSPE removes partially hybridized molecules. The step 7 is the detection, and the final step, which involves chemiluminescence or autoradiography, which results in dark bands in an X-ray film. If a radio-labeled probe was used, the bloat can be wrapped in a plastic wrap to keep it from drying out and then immediately exposed to a film for autoradiography. If it's a non-isotopic probe, then the blot must be treated with a non-isotopic detection reagent, prior to film exposure. Now, this is a diagrammatic representation of northern blotting diagram. Now, the applications of northern blotting involves, it observes a particular gene expression pattern between tissues, organs, developmental stages, environmental stress levels, pathogen infections, and other courses of treatment. The technique has been used to show overexpressions of oncogenes, downregulations of tumor suppressor genes in cancerous cells when compared to normal tissues. The gene expression, rejection of transplant organs, and the diagnosis of several diseases like Crohn's disease also can be achieved. The expression pattern obtained under given conditions can provide an insight on the function of the gene. Now, the detection of viral microRNAs also play a key role in viral infection, which can be identified by northern blot, and the variance in size of gene products which indicate deletion or error in the transcript unit and processing. By altering the probe target used along with the known sequence, it is possible to determine which region of the RNA is missing. And it can also finally be used to screen recombinants by detecting the mRNA formed by the transgene. This is a comparison between the southern and northern blotting. So the target of detection in southern blot, it's a DNA, and in northern blot, it's RNA. Now the sample prep, in both, it's the extraction of the needed nucleic acid. It involves enzymatic digestion, the deproteination, and here we use restriction enzyme in case of DNA, and in case of RNA, we use formaldehyde. Separation is done for both by gel electrophoresis, especially agarose gel electrophoresis. The membrane used can be both nylon, and the probe For DNA, that is southern blotting, should be a nucleic acid probe with single-stranded sequence homologous to the target DNA or the specific sequence we need. And in case of northern blot for RNA, it can be RNA, DNA, or oligodeoxynucleotides. The detection methods used for both southern blotting and northern blotting, that is DNA and RNA, can be X-ray film or chemiluminescence method. These are my references. Thank you.